Hello, my dear students. Welcome you all to Shri Jain Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with liver. Before continuing with this topic, in the last turn, we were discussing about the alimentary canal, different parts of alimentary canal. So, in the last, we have discussed the nerve supply. Okay. So the nerve supply, it includes basically Arabic nerve plexus and Meissner's nerve plexus. Okay, Arabic's nerve plexus, it is in between the longitudinal and the circular muscle layers and the muscularis layer. Okay, the nerve plexus between the longitudinal muscle and the circular muscle layer in muscularis layer, and the Meissner's plexus in the it is in between the submucosa and the circular muscle layer. Except in stomach, it is found in between the oblique muscles and the submucosa. Apart from it, I just want to tell you about, see, that autonomic nervous system which is present to control the peristalsis and the other activities related to the alimentary canal. Okay, so there are two types of systems of subsystems, I should tell, of autonomic nervous system. First is the parasympathetic, and the other one is sympathetic. Parasympathetic, it provokes the rest and digestion, and sympathetic, it is responsible for flight or fight responses. Like peristalsis and enzyme secretions are initiated by parasympathetic nervous system. Peristalsis and enzyme secretion is initiated by parasympathetic nervous system, right? Okay, and it is decreased by sympathetic nervous system. So it is initiated or increased by parasympathetic nervous system and decreased by sympathetic nervous system. By sympathetic. Okay, Arabic's nerve plexus, it controls peristalsis. It controls peristalsis. Okay, Meissner's nerve plexus controls the secretion from gastrointestinal glands. Meissner's nerve plexus, it controls the secretions from gastrointestinal glands. Okay, understood? Now, now we will talk about liver. Liver, it develops from endoderm. It is endodermal in origin. Weight is around 1.5 kgs. It function as both exocrine and endocrine. Okay. In human, it is found on the right side of abdominal cavity below the diaphragm. Okay. Below the diaphragm. The region of the right side of abdominal cavity is the right hypochondrial region. Hypochondrial region below the diaphragm. The liver is the largest gland of the body. It is the largest gland of the body. It is made up of left and right lobe. Okay. It is located beneath the diaphragm on the right hypochondrial region. It is the largest gland of the body. Okay. It is made up of left and right lobe. Left lobe is smaller than right lobe. Right lobe forms five five by sixth part of the liver. Okay. And left lobe forms 1/6 part of the liver. Right lobe forms 5 by 6th part of the liver, and left lobe forms 1/6 part of the liver. Okay. Liver is comprised of two lobes, okay, and these lobes are joined by falciform ligament. Right lobe and the left lobe of the liver are separated from each other, or they are joined by falciform ligament, which is a fibrous connective tissue. Okay, so falciform ligament is a fibrous connective tissue which is made up of fold of peritoneum, Okay, so liver is the largest gland. Okay, it is weight is 1.5 kgs. It functions as both exocrine and endocrine function. Although endocrine functions are very minimal, almost negligible. In human, it is found in the right side of the abdominal cavity below the diaphragm. Okay, so the region is right hypochondrial region. The liver is the largest gland of the body. It is made up of left lobe and right lobe. Left lobe is somewhat smaller than the right lobe. Right lobe forms 5 by 6th part of the liver, and left lobe forms 1 by 6th part of the liver. Right and left liver mean the lobes of the liver. They are joined by falciform ligament, which is a fibrous connective tissue. And this falciform ligament is made up of fold of peritoneum. It is made up of fold of peritoneum. Understood? This is the anatomy of the liver. You can see the right lobe and the left lobe. Okay. The right and left hepatic ducts, the right hepatic duct and the left hepatic duct. This is the right, this is the left hepatic duct. Then this is the cystic duct from gallbladder. Okay, so right and left hepatic ducts, they form the common hepatic duct. This is the common hepatic duct. Cystic duct merges with the common hepatic duct to form bile duct. This is the bile duct. So I will write here right and left hepatic duct merges to form common hepatic duct. Common hepatic duct CHD plus cystic duct from gallbladder forms bile duct. Okay, understood? Now this bile duct along with the pancreatic duct, okay, it merges in the duodenal region to secrete the bile salts, the bile pigments for the emulsification of fat along with the pancreatic duct which is carrying the enzymes, right? So each lobe of the liver is divisible into lobules. Each lobe is divisible into lobules, okay, which are structural and functional units of liver, which are structural and functional unit of liver. Understood? Okay. You can see the accessory pancreatic duct, duodenum, head of the pancreas. Okay. So right and left hepatic duct, they form common hepatic duct. Common hepatic duct along with the cystic duct from gallbladder. Cystic duct from gallbladder forms a bile duct. Okay. The right and left hepatic duct develop from right and left liver lobe. Both these ducts combine to form common hepatic duct. Gallbladder is situated below the right lobe of the liver. Okay, right lobe is big than the left lobe. Cystic duct of gallbladder is connected to the common hepatic duct to form a common bile duct, also called ductus choledocus. Okay, so ductus choledocus is the common bile duct, another name for the common bile duct. Common bile duct is formed after the merger of common hepatic duct along with the cystic duct. Okay, so the common bile duct is also known as ductus choledocus. Internally, liver is made up of numerous polygonal lobules. There are numerous polygonal lobules which are present, which are the structure and function unit of liver. These lobules are covered by Glissen's capsules. These are covered by Glissen's capsule. So Glisson capsule, we can say it is a fibrous sheath beneath visceral peritoneum. beneath visceral peritoneum. Okay, this fibrous sheath beneath visceral peritoneum is known as what? Glissen's capsule, which covers the lobules of the liver. Okay, it surrounds the lobes and the lobules of the liver. Liver is further made up of right lobe and the left lobe, as already discussed. The right lobe, it is further divided into right lobe is further divided into right lobe proper, caudate lobe, quadrate lobe. So right lobe is further divided into right lobe proper, caudate lobe, and quadrate lobe. Okay. Each lobe consists of radial rows of hepatic cells. Each lobule consists of radial rows of hepatic cells, which are called hepatic cord. Each row is each row is one or two cell wide and two cell thick. In between the hepatic cord, a space is present, which is known as what? Hepatic sinusoid. Okay, these sinusoids are filled with blood. They are filled with blood. Okay, so it is like the right and the left hepatic duct. They form the common hepatic duct. Common hepatic duct forms the along with the merger of cystic duct from gallbladder forms the common bile duct, which is known as what? Ductus choledochus. Internally, liver is made up of many lobes and lobules which are covered by Glissen's capsule. Glissen's capsule is a white fibrous sheath, okay, beneath visceral peritoneum. Each lobule, when lobes are further divided into lobules, each lobule consists of radial rows of cells which are called hepatic cord. Each row is one or two cell wide and one or two cell thick. In between a hepatic cord, a space is present, which is known as what? Hepatic sinusoid. These sinusoids are filled with blood. So this is about the internal and the external morphology, internal anatomy of the liver. This now coming to the anatomy of the liver. The sinusoids, which are lined by endothelial cells mostly, but a few macrophages cells are also present. These are called Van Kupfer cells, which are phagocytic cells. So sinusoids, they are lined by the endothelial cells. They are lined by the endothelial cells, but a few macrophages. There are some macrophages, okay, or macrophages cells which are present. These macrophages, in case of liver, they are known as what? Van Kupfer cells, which are phagocytic in action. Okay. The bile canaliculi, okay, the bile canaliculi run in between the two layers of the cells in each cord. Hepatocytes, which are the hepatic cells, pour bile into the canaliculi. Canaliculi open into the branch of hepatic duct, which is situated at the angular part of the lobule in the Glissen's capsule. All branches of hepatic duct of right and left lobe are combined to form right and left hepatic duct, which comes out from the liver and forms a common hepatic duct. Okay, so the bile canaliculi, they run in between two layers of cells, okay, in each hepatic cord. Hepatocytes, these are the cells or the hepatic cells which pour the bile into the canaliculi. Canaliculi open into the branch of hepatic duct. It opens into the branch of hepatic duct, which is situated at the angular part of the lobule in the Glissen's capsule. All branches of hepatic duct of right and left lobe are combined to form right and left hepatic duct, which come to form the liver and forms a common hepatic duct. Hepatic artery and hepatic portal vein enter into liver and divide to form many branches. They enter into liver, hepatic artery and hepatic portal vein. They enter into the liver and divide to form many branches. These branches are also found at the angular part. They are also found at the angular region at the angular part. Its fine branches open into the hepatic sinusoids. Further, these branches they open into hepatic sinusoids, branch of hepatic portal vein, branch of hepatic artery, and branch of hepatic duct. They are known as what? Portal triad or the hepatic portal triad. Hepatic artery and hepatic portal vein, they enter into liver and divide to form many branches. These branches are also found at the angular part. Its fine branches open into the hepatic sinusoids, branch of hepatic portal vein, hepatic artery, and branch of the hepatic duct. They are also known as portal triad. All hepatic sinusoids open into central vein or intralobular vein through fine aperture. All hepatic sinusoids, they open into central vein or intralobular vein through fine aperture. All central vein combine to form hepatic vein. All central vein they combine to form hepatic vein, which comes out from liver and opens into inferior vena cava. All central vein comes, all the central vein they combine to form hepatic vein. All the central vein they combine to form hepatic vein, which comes out from the liver and opens into inferior vena cava. So the inferior vena cava receives the hepatic vein. Hepatic vein basically it is a merger of all central veins. Okay, all hepatic sinusoids open into the central vein or intralobular vein. So intralobular vein to hepatic vein to inferior vena cava. Okay. I'll repeat once again. Sinusoids, they are lined by the endothelial cells along with macrophages known as what Van Kupfer cells, which are phagocytic. The bile canaliculi find, slit-like apertures which run in between the two layers of the cells in each cord. Okay, hepatocytes of the liver cells, they pour bile into canaliculi. Canaliculi open into the branch of hepatic duct, which is situated at the angular part of the lobule in the Glissen's capsule. All branches of hepatic duct, they merge to form the right and left hepatic duct, which come out of the liver and forms a common hepatic duct. Hepatic artery and hepatic portal vein enter into the liver and divide to form many branches. These branches are also found at the angular part. Its fine branches open into the hepatic sinusoids. Branch of hepatic portal vein contains the branch of hepatic portal vein, hepatic artery, and branch of hepatic duct. They are known as what? Hepatic sinus, hepatic tride, portal tride. Hepatic sinusoids they open into central vein or intralobular vein. From intralobular vein to the hepatic vein and hepatic vein to inferior vena cava. This is the histology you can see. The hepatic lobules is the portal tride, bile duct which is green in color, portal vein which is blue in color, hepatic artery which is red in color. Okay, these are the sinusoids in the spaces. You can see the sinusoids. The central vein or the hepatic vein, hepatocytes, von Kupffer cells. Understood? Von Kupffer cells, which are known as what? Phagocytic cells, also known as phagocytic germ cells. Phagocytic germ cells. There is a presence of pre-sinusoidal space, central vein. Okay, yeah, this is the liver. Sinusoid, septal branches, Kupffer cells, hepatocytes. Okay, they merge to form central vein. Okay, all these merges to form central vein. Understood? This liver, inferior vena cava, which receives the hepatic vein. Hepatic vein, it comes from the central vein, and central vein comes from different hepatic portal veins. Okay, hepatic artery, gallbladder, sinusoid, blood-filled space. Functions of liver. Most of the biochemical functions of the body are done by liver only. Secretion and synthesis of bile. This is the main function of the liver. Bile is yellowish, greenish in color. It is alkaline fluid. In bile juice, bile salts, sodium carbonate, bicarbonate, glycolate, taurocolate, bile pigments, cholesterol, lecithin are present. So in case of bile juice, bile salts, bile pigments, sodium bicarbonate, sodium taurocolate, sodium glycolate, okay, cholesterol, lecithin, all are present. Bile salts help in emulsification of fats. Bile salts they help in emulsification of fat. Okay, mean breakdown of bigger droplets of fat into smaller droplets. Bile prevents the food from decomposition. It prevents the food from decomposition. It kills the harmful bacteria. Carbohydrate metabolism. The main center of the carbohydrate metabolism is liver. Following steps are involved in the carbohydrate metabolism. Glycogenesis, glycogenolysis, and gluconeogenesis. Glycogenesis is the conversion of Yes, conversion or storage of extra amount of glucose into glycogen from the digested food is called glycogenesis, mean formation of glycogen. Glycogenolysis, the main stored food in the liver is glycogen. Glycogenolysis, the conversion of glycogen into glucose back in glucose level in the blood falls down. Gluconeogenesis, at the time of need, liver converts non-carbohydrate compounds into glucose. This conversion is called gluconeogenesis, meaning formation of glucose from non-carbohydrate sources. Okay, this is called neoformative process of glucose. Glycogenolysis, synthesis of glycogen from lactic acid, which comes from muscles, is called glycogenolysis. Glyconeogenesis. Okay, bile juice, it contains water and bicarbonate ions along with bile salts. Okay, then bile pigments, bilirubin, biliverdin. Okay, bile pigments are the result of the breakdown of products of RAC, the RBC, sorry, bilirubin, biliverdin. They all are having almost same colors. So glyconeogenesis, gluconeogenesis, glycogenolysis, and glycogenesis. Okay, these are the terms. Storage of fats. Liver stores fat in the small amount. Hepatic cells an important role play in fat metabolism. The storage of fats increase in the liver of the alcoholic addict person. Fatty liver. This storage of fat increases the activity of liver. The damage of liver due to the alcohol intake is called alcoholic liver cirrhosis. Deamination and urea formation. Deamination of amino acid mainly done by liver. Amino acid ammonia. Separation of urea from amino acids is done by liver. So Krebs-Hensley cycle. Formation of urea. Formation of urea, deamination of amino acids, everything is done in liver. Okay, liver converts ammonia into urea through ornithine cycle. After this, after the spoilage of the liver, the ammonia level in the blood, animal body increase and the animal dies. It dies. Okay, so deamination, urea formation, hydroxylation, all are the functions of these bile juices, the liver, and the main functions of liver. Okay. Purification of blood, spleen and liver separates dead blood cells and bacteria from blood. Okay, spleen and liver, they separates the dead blood cells and the bacteria from the blood. Kupffer cells in liver and phagocytes in spleen performs this function. Kupffer cells in liver and phagocytes in spleen perform this function. Purification of blood. The spleen and liver separate dead blood cells and bacteria from the blood. Synthesis of plasma proteins. Many types of proteins are present in the blood plasma. All are proteins except gamma globulins, which are synthesized in the liver. Gamma globulins are not synthesized in the liver. Gamma globulins are the antibodies which are formed by lymphocytes. Most of the factors which are responsible for clotting of blood are synthesized in the liver, means clotting factors. Synthesis of heparin. Heparin is an anticoagulant which is a mucopolysaccharide. Heparin is an anticoagulant, which is a mucopolysaccharide. Heparin is also formed by basophils. Okay, that is a special type of white blood cells. So synthesis of heparin. Heparin is an anticoagulant. Most of the factors are responsible for clotting of blood. Okay, that's synthesis of vitamins. Liver changes carotene into vitamin A. Beta carotene is a photosynthetic pigment which is obtained from plants. It is abundantly found in carrot. Okay, liver stores vitamin A, D, E, K, vitamin B12, storage of minerals. Liver stores iron, copper, zinc, cobalt, molybdenum. Liver is a good source of iron. Liver is a very good source of iron. Detoxification, the conversion of toxic substances into non-toxic substances done by liver. The toxic substances are formed by metabolic activities of body. For example, persuic acid is converted into potassium sulfocyanide. It is a non-toxic salt by the liver. So persuic acid is converted to non-toxic salt by the liver, potassium thiosulfocyanide. Hemopoiesis, it helps in the formation of blood cells. In embryonic stage, RBC and WBC are formed from liver. Yolk synthesis, most of the yolk is synthesized in the liver only. Secretion of enzymes. Some enzymes secreted by liver participate in metabolic functions, fats, carbohydrates, metabolism, dehydrogenase, cytochrome oxidases. Prothrombin and fibrinogen are also formed in hepatic cells. These help in blood clotting. Prothrombin and fibrinogen, they are also found in hepatic cells. These help in blood clotting. Factors 1, 2, 5th, 8th, 7th, and 9th and 10th are formed in liver, which are responsible for blood clotting. Factor 9, factor 10th, factor 7th, factor 5th, factor 2, factor 1, all are synthesized in liver for blood clotting. Prothrombin and fibrinogen proteins are also formed in hepatic cells. They help in blood clotting. Secretion of enzymes is done. Okay, york synthesis is done, hemopoiesis is done, detoxification is done, storage of minerals done. Okay, so that's it for today. I mean, for liver. Okay, we will be starting with pancreas in the next one. Till then, bye bye, take care, stay safe, stay healthy.